today's lecture we will continue with our study of robustness in biological systems, and we will study the different mechanisms underlying robustness, namely system control, redundancy or diversity that contribute towards robustness, and modularity and decoupling, as well as hierarchies and protocols that are existent in biological systems that enable robustness. So, let us continue looking at the mechanisms for robustness in biological systems. So, we did talk about backup or fail-safe systems or redundancy, then disturbance rejection through feedback control, decoupling, modularity, and you can have decoupled modules, but then they need to be coordinated. How do you coordinate these modules and so on? You need to establish certain hierarchies and protocols. So, how do these things come within different kinds of systems, be it engineering systems or more interestingly for us here biological systems? How do you coordinate these various systems? So, you need to obviously control these systems, and that is typically via some negative feedback or positive feedback. So, how does negative feedback work? So, some disturbance is there and you compensate for that disturbance, right. So, for example, that happens all the time in chemotaxis and so on. So, you have a system that is a bacterium that is randomly rolling, tumbling in some direction, and then there is it senses that there is some other signal coming in from a different place. So, you need to adjust your current direction of current vector of movement towards the gradient and move towards that, right. So, that is a response, it is a feedback to the based on the new signal that has come in, right. You can also have positive feedback which contributes to robustness by amplifying stimuli, right, and typically positive feedback will give rise to a sigmoidal kind of function, or more ideally a step function wherein you have a bistable system wherein you are either at an off state or at another on state wherein you are stable, right. So, this contributes to robustness by amplifying stimuli, often having bistability, so that the activation level of a downstream pathway can be clearly distinguished from non-stimulated states, right. So, that you can either maintain it at off or at on, and this happens in, for example, Drosophila segment polarity and so on. You can read a little more about this, I will share the paper with you as I was talking about, but basically you have two types of feedback, both negative feedback and positive feedback, which help you maintain robustness, right, and there is a combination of all these that happens in any given biological system. So, then you need both redundancy and diversity. So, what do you mean by, you understand what is redundancy, right? What is diversity? Maybe in biological terms one would call it heterogeneity, right. So, you do not have two homogeneous things doing the same thing, but two reasonably different ways of doing the same thing, because if you have two identical things there is a good chance of having a what is known as a common mode failure. It is something that is, if there is a perturbation that A is susceptible to, B might also be susceptible to the same perturbation. So, if you have two very similar proteins catalyzing the same thing, the same reaction, suppose there is a temperature heat shock, both proteins may get denatured, right. So, you have not availed of any of the advantages of the redundancy. By diversity you have a different way of doing the same thing which is very likely different from the original way. For example, the equivalent on this is what you have on an aircraft. So, you have the same entity that is being computed by two different algorithms. So, if algorithm 1 fails or if computer 1 fails, computer 2 is need not fail, right, but if you have two computers that are identical, both might actually fail. This is something that one often worries about while setting up hard drive arrays, right. You might have all heard of RAID arrays, right, redundant array of independent disks. So, the thing is you usually put same types of drives in those arrays, and you make redundant copies, so there are many RAID levels and so on, let us not get into those details, but maybe there is the same manufacturing defect in both of them. So, you are much better off like putting two different branded drives of the same size rather than two identical drives from the same batch. Maybe there is the same bug, right, both of them fail after 7 months of operation, right. So, that is not diverse, right, it is redundant but not diverse. By diversity you would want a completely different type of drive, right, maybe even a slightly larger drive from a different manufacturer in a different batch, which is very likely not going to have the same average failure rate as the original one, right. So, robustness can be enhanced obviously if there are multiple means to achieve a specific function. So, failure of one gene or one enzyme can be rescued by some other substituting enzyme of gene. An alternative or fail safe mechanism is usually attained by having multiple heterogeneous components and modules with overlapping functions. So, you have module 1 and 2, maybe module 2 and 3 can take over together for 1 and 2, right, or maybe 2, 3 and 4 will do the same job as 1 and 2, right. So, you can have different how does this happen in biology? This happens normally by gene duplication, right. So, this is a very common process that happens in most especially very heavily in smaller organisms. So, yeast has had several gene, your Saccharomyces cerevisiae that you see today as a result of several gene duplication events that have happened in the past. So, you have had something known as a whole gene duplication event as well as a small scale gene duplication event. So, either a bunch of genes get duplicated or a whole, the entire genome gets duplicated. Because of this, there is a lot of redundancy that can happen. So, initially if you had an essential gene, you had only one copy of it and there was no latitude for that to mutate and so on. Now, if you have two copies, they can slowly diverge in function. Maybe this gene can still do the same thing as it was doing all along, whereas this gene can take on some allied functions which can be very useful in say a different environment and so on and so forth. And alternative mechanisms are obviously coupled with control to ensure robustness. Maybe the cell will not express both genes at the same time. If you have isozymes, it will express only one of them, and when the first one goes off, it will start expressing the next one, right. So, there is a control mechanism that underlies this whole robustness aspect of the cell. And the existence of alternative mechanisms allows regulatory feedback to remain intact even though there are several perturbations. And you need very specific regulatory networks to achieve this, because you need to control across several, maybe there is a metabolic layer, there is a regulatory layer, you need to transcend across these levels to control the cell carefully. The other very interesting mechanism is one of modularity. So, it is an effective mechanism for basically insulating certain perturbations. So, you have one module, maybe only that starts failing, or a mutation only affects that module. This is intimately connected with decoupling as well, right. Modules are somewhat decoupled, this minimizes effects on the whole system. So, you find it widely, be it biology or in engineering, right. You always have plug and play modules, right, and if a few classes down the line we will talk about synthetic biology where this is a vision, right. You want to have plug and play modules. I put an oscillator into the cell, it just works, right. I plug it into the rest of the modules that are already existing in the cell, it starts working as we imagined, right. And you can have different types of modularity: functional, spatial, temporal, right. I think what is spatial modularity? The easiest of them, like compartmentalization. Functional modularity is two different functions, it could be even redundant, but two different modules trying to do different functions or same functions. Temporal is one module is active at one time step, the other module only happens at a very different time step. So, they do not overlap in time. So, modules do not overlap in time, space or maybe function. So, decoupling. So, this isolates low level variation from high level functionalities. So, if a system is well integrated without modular structure, any small change in one part of the system might have very far reaching changes in other parts of the system, and this is problematic. So, you might have a a small noise might give you a very bad outcome and so on. Next interesting part is that of hierarchies and protocols. So, how do you orchestrate this within the cell? So, protocols encompass a set of rules underlying all the relationships between the different entities, right. So, what is the language within the cell? Usually some sort of small molecules that transfer signal, right, or ATP and so on, and there is an organizational structure in which these modules are embedded. Let us see what it is. And you also need some currency for system function like ATP or NAD, some energy molecules, protons and so on, and this facilitates a sort of layered regulation. So, if you see any signaling cascade, you will see that there are several layers at which you can effect control, and there are molecules that transmit information across these layers. So, in today's video we looked at some of the interesting mechanisms that enable robustness in biological systems such as system control, redundancy, modularity and so on, and in the next video we will start looking at the organizational principles or the key structural features that enable robustness, basically a bow tie structure.